Hi, my name's Tabitha King. I'm a PhD student studying environmental science and public policy. I actually did my undergrad at Mason as well, studying environmental science and conservation. I've been a grad student at George Mason for about 6 years now. I'm actually kind of wrapping up my PhD. I have a year and a half left. My current research looks at per- and polyfluoroalkyl substances, or PFAS, in aquatic organisms from the Potomac River. So far, I've collected things like submerged aquatic vegetation, plankton, small fish, larger game fish, and surface water and sediment. Through all of that, I've both been out on a boat collecting the samples, and then I come back to our SciTech campus or the Potomac Science Center to process those samples. And then the final sample extracts are then run on this instrumentation that we have at the Potomac Science Center. It's a liquid chromatograph mass spectrometer. With that, we're able to target 39 different PFAS compounds to see their levels in the organisms and if there are any spatial patterns from where they were collected from. And this work's important because there's still a lot of knowledge gaps in how PFAS are accumulating in aquatic environments. And also for game fish, there's a lot of anglers or local fishermen that are relying on these species to maintain their own personal or household food security. Looking at the PFAS levels in these game fish species will help us provide recommendations for agencies like the Virginia Department of Health on whether or not they need to create fish consumption advisories, because right now there are currently no PFAS fish consumption advisories in the state. Along the Potomac River, there's only advisories for things like PCBs and mercury, but because of how much PFAS bioaccumulate in fish, it's really important to know the levels in our local fish in case these provisions need to be put in place to protect angler health.